Crossover. There are two widely used genetic operators. So crossover is one of the two widely used genetic operators. Crossover is also known as recombination in the literature. So sometimes the terminology recombination will be used synonymously in the literature. Crossover shows that offsprings inherit characteristics from their parents by swapping different parts of their chromosomes. By swapping different parts of their chromosomes, new combinations of characteristics can arise in the offspring through this crossover operator. The newly created offsprings are randomly paired, and parts of their chromosomes and parts of their genetic materials are swapped, and thus due to this genetic swapping of the homologous chromosome, new recombinations arises which imparts new properties to the individuals, which are expected to be more suitable, more fitted as compared to their previous generation. So they are swapping their parts with some probability, which we can denote in these genetic algorithms with probability of that C. Crossover operators comes in different form. There are different forms of crossover operators. For example, in this figure, we can see that this figure is representing and highlighting different forms of the crossover operator, and through this figure, we will try to understand the basic logics and the mechanism of the crossover, different types of crossover operators, different forms of the crossover operators. This figure A is representing 1-point crossover operator. So crossover operator may be of 1-point crossover if it is taking place at just a single point. There may be a 2-point crossover recombination as well. If recombination is taking place at two different points of the homologous chromosomes, it may be of uniform form. We will discuss what is a uniform type of crossover. It may be of arithmetic form. There are some crossover forms which are specialized for the sequences. So for sequences, and there are some forms which are specialized to be used for data representation in the trees specifically. So first we are going to highlight what is a 1-point crossover. In 1-point crossover, chromosomes are going to exchange their parts at a single point. For example, here we can see that at this point both the chromosomes are exchanging their parts. So what will happen here, that this chromosome from here it will be original, but from this point onward this part will be swapped with this one, and this chromosome will take the part of this chromosome. Now the next combination will be like this, that in this new combination this chromosome has this original part, and this is the newly recombined part with this chromosome, and this second chromosome has this original part here, and the newly swapped part is here. So this 1-point crossover will yield two new chromosomes for the next generation, which has swapped their parts on one specific point. Now the second one form of the crossover is a uniform crossover method. In uniform crossover method, at specific uniform points, the chromosomes are going to exchange their parts. For example, in this case, this chromosome is exchanging part here, and then here, and then here, and then from a very uniform point it is going to exchange parts here, and similarly same is the case with this chromosome, and it will swap parts here and here as uniformly as the other one has taken place. And so here we can see that this chromosome is now in this form, and this chromosome is now in this form, have swapped their parts uniformly throughout the length of the whole chromosome. The third one form is the arithmetic form of the crossover. In arithmetic form is used where the data is not represented with in the discrete manner. These two chromosomes will yield a single chromosome through arithmetic mean. For example, here is the value is 0.2, and here the value on this chromosome at that specific locus is 0.4. The arithmetic mean of 0.2 and 0.4 is 0.3. So it will yield a new value of 0.3 here, and similarly 0.6 and 0.4 will yield an arithmetic mean of 0.5. So here we can see that the new value is 0.5 on this locus. And similarly, at all the loci of this chromosome, the values at these specific loci will yield its arithmetic mean to yield a new chromosome for the next generation. Similarly, another form of the crossover is crossover for sequences. And in this, where both there are two chromosomes here, and they will yield us a specific chromosome by shuffling their parts. For example, the new chromosome will take this part from here, BCD, and this selected part from here. This E is now extra from the total length of the chromosome, so it is out over here, but this E can be now shuffled over here, and now the next chromosome will have a combination of E, B, C, D, G, F, A instead of either this one and this one. Similarly, there is another type of the crossover which are used for tree representations. This is specifically a tree representation over here. So we have one chromosome represented in a tree over here in a dark color, another one with the dark color in the light color here. Now these two will exchange parts here in the tree and here in the tree. So this will yield its original over here, but from here it will be a new one. So we can see that there is the original part over here, but this part is being swapped with this one, and this will yield, it's retaining the original, the crossover point is over here. So this part has been swapped with this chromosome here, so it is taking the part of this chromosome, taking this part of this chromosome, and this is taking this part of this chromosome. So crossover operators can be applied either to the chromosomal representation, the genetic representation, either it is in the form of linear chromosomes or they are in the form of trees. In either case, crossover operators can be applied successfully, and it will yield to new genetic combinations of the novel chromosomes for the next generation.